Hello again, everybody. Welcome back to our MIDD Plus 2023 Clinical Track program. This is a Q&A section. We really appreciate all the speakers came back now on the screen. And really appreciate all of your presentations previously. We opened the Q&A tab for the audience again. So please, if you have any questions, please feel free to type in using the Q&A in your dashboard. And I would like to remind everybody again that by participating in this sessions, you allow us to contact you for follow-up questions as we treat your privacy very seriously. Secondly, is that please feel free to ask questions, we really want to have this session as a very interactive one. So,
is it only me, or am I hearing an echo or a delay?
Anybody else hearing the echo?
No.
Looks okay on my end.
Okay.
Yeah.
Okay. Thank you. And so maybe I will provide a couple of questions started, and please, I probably point to a specific speaker to initiate a conversation, but I encourage everybody to jump in to address if you have any comments or thoughts. And first one, I will direct it to Sandra first. And so in your presentation, you mentioned that the regular agencies sometimes open the possibility using surrogates in lieu of preclinical data or early development data. And so could you kind of talk about that, how this could possibly impact the use of modeling simulation and MIDD platform? This is probably related to what Raj has presented. So I would ask you to go as a second one, that to comment, but Sandra, based on your experience, could you share some thoughts with us? You're muted, Sandra.
Sorry about that.
Sure.
I was saying that I think you're all familiar with FDA, that FDA has a long history of promoting the development and use of new technologies, including how to better predict human and animal responses. FDA formed a working group called the Alternative Methods Group, I think in 2020. And I'm sure that Raj is familiar with that. And as part of this working group, there is a webinar series initiative inviting companies and developers to showcase their cutting-edge technology to FDA scientists. And these cutting-edge technologies include the use of model and simulation for first-in-human predictions. FDA also encouraged the publications of any alternative technologies, and also these papers can be co-authored by FDA scientists. So hopefully with this background that I'm giving to you, it's clear that QSP and QSRP, PBPK have a great potential to support the use of surrogate, to be used as surrogate for preclinical studies.
Mm-hmm. Thank you. And Roger, do you have anything to add based on you've seen the people approach agency with different programs and focus on the preclinical translational to early clinical? What's any thoughts that you could provide?
Sure. I think Sandra laid it out nicely. I think there is a long practice from the agency side to be able to most efficiently and effectively utilize all the available information. And that wouldn't be any different as it pertains to the use of biomarkers in past activities in this particular space. So there is a rich history. For example, in PDUFA 6, there was a specific, if you notice one of my slides, it was about biomarker qualification and development, along with MIDD and several others as regulatory development tools. The the aspect about alternative technologies that Sandra pointed out is definitely something that agency is looking forward to be able to see how they can be incorporated to advance the development. I think everything depends upon the quantum of information that is available, the context in which it is being utilized. We all get excited when we hear the word surrogate, and then it almost feels like it can replace registration trials. But if we are only focused on that, I think we are shortchanging ourselves, because drug development is just not one phase 3 trial. It is leading up to the trial and afterwards also in the life cycle. So there are so many opportunities for a fit for purpose application of biomarkers, and agency has been very much open to exploring ways to identify in what context how these can be applied. And along with those modeling and simulation approaches, there is also a focus on other approaches, such as the microphysiological systems, for example, as a ways to help explore the use of these technologies in the preclinical kind of a stage or early translation phase. I'm pretty sure when we connect all these dots together, the more systems-based approaches are going to become more and more powerful in helping advance drug development.
Great, thank you. And so the next two questions were directed to Raj, and so one of the questions actually came after we right after we drop off the call, but it's really important and interesting one. So in your slide, you show that in 2022, there's approximately 50% of the projects proposed to the agencies got denied. Two related questions to it. So what happened to the program? It's the submissions being denied. And secondly, besides agencies' resource consideration, is there any key features that you've seen in the past that the sponsor could have done a better job to support their request and becoming a stronger package for the agency to consider? Thank you.
Sure. Excellent observation by who made that. It couldn't have been any more stark. But I would start off by saying, as we approached that 5th year of the program, we were really seeing a lot of strain on our bandwidth. Initially, we wanted to make sure we had the appropriate experience that we wanted to gain with the program, because we were standing up an entirely unique regulatory engagement pathway. We did not have a lot of experience to look back to see how this can be set up. So we definitely put way a lot more than what our capacity was in the initial phases to make sure that we were able to pressure test the system, see what are the limits of our bandwidth. But as we were progressing towards the later phases under PDUFA 6, we really started seeing the constraints on the bandwidth, and it almost became unsustainable. So that's the reason why you would see that you would see close to around half of them were denied. But not all of them were because of purely bandwidth. But bandwidth did contribute significantly in that particular year, or it started actually coming from later half of 2021, but it showed up really here. In addition to that, your second part of the question is, how can people make strong packages, right? And part of our denials, as I had described, were also those instances where people have in mind and probably they have not thought through how to best position that application for a regulatory decision at hand. So sometimes is not full thinking through of what kind of application is going to be there, for example. The other instances are, you are now asking for the moon kind of thing. So you're coming up and saying, this is an entirely new biomarker, which we will use for seeking approval, for example, without having the background. Obviously, we cannot engage in full-blown regulatory research kind of activities during these interactions. These are meant to actually address ongoing programs. So if someone were to come with a novel marker or a novel response measure, it becomes a several years project essentially to be able to resolve the robustness of such a finding. So it goes beyond the scope, for example, or it's
Sure.
almost like qualification of an entirely new marker for which there are alternative pathways that we generally direct them to those. And lastly, there are some unique instances where there are outstanding regulatory issues, which there are engagements with the clinical divisions wherein a clear expectation has been set, which is a minimum requirement. And sometimes people pressure test to see whether they can overcome those using some alternative approaches. And sometimes the areas are so nascent, it becomes challenging from a regulatory perspective, that the uncertainty is so large that we will not be able to overcome the prior expectations that have been set forth in such nascent areas. If these were areas where there was a lot of maturity, definitely we encourage the utilization of MIDD approaches. But there are some situations which are rapidly evolving and there are prior expectations. So we would definitely like to make sure that we are not using MIDD to bypass prior expectations. We want them to be complementary and supplementary, such that at the end of the day, our major objective is to decrease the residual uncertainty while efficiently making the decisions. So those are some of the elements. I hope I addressed aspects of your question.
Certainly, thank you. And maybe one more question before we move on to other speakers is that based on your presentation, very clear that at least at the initial stage that the focus scopes are in 3 topic areas, right? Dose selections, optimization, clinical trial simulations, and mechanistic safety evaluations. And so is there a plan under the PDUFA 7 that could be expanded even more? Or what is agency's plan in general in terms of further focus on the 3 areas, or what's a future plan at this time?
Yeah, I could have made it a little bit more clearer during my presentation. Yeah, we started with those 3 as initial priority focus areas. The reason was it was a combination of areas where we had prior experience, so we knew we could deliver on, and there were areas with emerging applications where we thought we are going to get a little bit more experience as part of this pilot. As we got more comfortable, almost I would say second half of 2021 leading up to 2022, we did not see these areas as only those areas where we would still be focused. We actually started expanding. People came up with other applications such as bridging, for example, PK bridging kind of approaches are very similar to the last presentation that we heard, using exposure response analysis for slightly different indications and things of that nature. We started branching out. So what we think going forward in PDUFA 7 is we are not just limiting ourselves to these areas. We would be open to any areas as long as they are within the scope. And if you were to remember our definition upfront, we would definitely consider that those which are applying some complex innovative designs, trial designs kind of aspects, or those which are involved in operating characteristics of trials or sample sizing kind of those for phase 3 trials, which predominantly fall within the statistics domain or the complex innovative trial designs. We would be open to any kind of applications. We have had applications utilizing PBPK models for alleviating the need for certain drug interaction studies, or trying to better inform based on whatever preliminary information that is there.
Mm-hmm. Great, thank you, Raj. And so the next question I would like to direct to Manuel. And so very nice presentation. You show us great utility using the Monolith suite in terms of building up a platform, and the clinicians, even more pharmacists or pharmacy could be benefit by looking into different variables and simulated it out. So, I mean, for the model-informed precision dosing per se, that have you had a chance to compare other software tools or other methods, maybe even more established, and so I guess I'm asking you that that focus on use this platform. Can you give us a little bit more introductions or your thoughts on that?
Thank you, Tom. That's a great question. I am. Yeah, of course, moving on to the post-marketing approval and all the challenges we have to implement pharmacometrics in the clinical setting. There's a lot of variables and different challenges that have made, as you are aware, that pharmacometrics establishment in the clinical setting in a hospital, so at the bedside, is still an ongoing process. And that's been evolving. There are different reviews on the software that is available to implement pharmacometrics for precision dosing, and it's definitely a continuous evolution during the last 10 years or so. Although there are software that has been available for more than 20 years, but it is definitely so. The point of these softwares is that these tools are really promoting or pushing forward the field, so the implementation or the reach of pharmacometrics into the clinical setting, and that's very valuable. They also exploring different technical and methodological solutions to better predict and to be more accurate and precise at the time of predicting the pharmacokinetics and pharmacodynamics. So, but still, we have a lot of challenges to implement those softwares in a hospital. And I would like to slight differentiation. You can have, so there are, for instance, here in Latin America, in different countries, but I know a lot of colleagues in Chile which use a particular software that has been expanded and promoted in Chile, but they are not pharmacometricians implementing MIPD, but they are implementing MIPD. Mm-hmm. And that's definitely I think we need pharmacists involved in TDM, in therapeutic drug monitoring, being able to perform all the methodological actions behind MIPD to bring pharmacometrics into the clinical settings, despite them being not involved in the technical aspects of pharmacometrics. But there are another groups, such as our group, which are developing pharmacometric models and doing research in the field, and also teaching and training new pharmacometricians. And so as we are developing models, we have kind of another needs, and we are trying to promote from a different point of view. So the solution that we developed is more suited to our needs in terms that it's very flexible, and we can use any model we develop or we validate in our population to start doing model-informed precision dosing, which is for us very useful, because if we use available software, we will have to pick different models which are already integrated into these tools and validated. They are validated, but in different populations. And as in my presentation, it's very important to validate the model in the population that will be in the targeted population, and that can be very challenging. And so available software is great. There are very different tools. There are a free tool, for instance, TD Metrics from the Sebastian Switcher group. And these groups are definitely leading the field. We also follow them and have tried some of their solutions, mostly TD Metrics. But when it comes to apply any model we want, even a PEPK or maybe a QSP model, this tool provides us more flexibility to start implementing and giving dosing recommendations to the clinical team here at the hospital.
Great, and based on your descriptions, also mentioned about other Latin American countries that with similar effort with different progress and things like that. Have you you were prolific in terms of publishing in this area and interact with different investigators. How do you see other regions other than the Latin American countries at this time? What's your observation at this point using this kind of approach?
Yes, so pharmacometrics in Latin America is really it's a very incipient field. I think that from my perspective, the reason behind that is that pharmaceutical companies in the region are not involved or are not that involved in bringing new drugs into the market. So they're mostly dedicated to the generic market. But during the last years, we are seeing a lot of improvement, a lot of progress of pharmaceutical companies implementing PBPK and PBBM for formulation for drug development, which is great, and that is happening like really fast. And we are promoting that from a group of an international group, which is the Ibero-American Pharmacometrics Network, REDIF, which we have done a lot to promote the field in the region, but still in the clinical setting is challenging.
Yeah.
Yeah, it's very hard when physicians and authorities are not knowledgeable about the field, about the advantage of implementing this type of approach. So it's definitely we have to move to. The great thing is that we can things that are already being done in other countries where pharmacometrics is more advanced and established. And so we are working in that direction, but a lot of things to do. And I think at REDIF, this international network has been key to in that sense, to promoting pharmacometrics here in the region.
Great, thank you, Manuel.
You're welcome.
And so my next question would direct to Naveed. That very nice presentation for Lena Capovel. And so you have shown a very specific example using Gestra Plus to build a model in the translational features between rat and healthy volunteer with a subcutaneous administration. And so the question back to you is that can you share a little bit about selecting rat as your preclinical model? What are the advantage or challenges by selecting this species, and is there any thoughts originally that with different selections of animal species like a mini pig or others, and so how and contributions that modeling approach Gestra Plus also that facilitate that process for you to perform the selections and modeling exercise. Thank you.
Sure. Thanks, Tom. Nice question. We just focused on one of the species, so we have data from multiple species, and as I mentioned during my talk, it's a work in progress, so we are working on other species as well. So based on the availability of the data and the aspects what we have developed so far, we have just shown with the rat. There is a future work or ongoing work where we will expand it with the other species as well. So yes, that is going to come.
Great, thank you.
Thanks.
The next question will direct to Viera. That very nice overview about the DDI aspects of modeling, and so typically the people will have questions that, can I have access to the standard models, files, or documents in the system? And if there is something that more complicated in terms of dynamic DDI assessment, that what's your recommendation for a scientist in this area looking at PBPK to build that kind of model? And so can you talk in a general terms for these questions? Thank you.
So when it comes to the general applications for the dynamic DDI, the DDI itself is actually fairly simple. Doing the DDI prediction, it's the simplest part of that process. The more critical part is building the baseline model. But again, if this is something that is incorporated as part of your model development process, starting early on, and as you are getting information from preclinical, from clinical studies, and so on, and continue improving the model, then you do have a model ready to start doing DDI predictions. So it doesn't have to be a difficult task, but the accurate prediction of DDI is really going to depend on having a good model for your victim and your perpetrator compounds to be able to capture the DDI. So that is the most critical part there.
Great. Thank you. I'm looking at people submitted question online. This is one, go right back to Raj. So the question is that, have you seen any requests to use MIDD to generate new data to support any clinical studies that run into enrollment difficulties? Recall that during the beginning of 2020, we have a COVID pandemic situation, and so it will be difficulty situation for enrollment and have patients to visit clinic to get blood samples or any clinical assessment. And so does the agency have any experience for a sponsor approach you for those kind of questions, using more innovative modeling approach to support sampling time, or to have a bit more different ways to handle PK/PD exposure response analysis for dose selection and justification? Thank you.
It's a very insightful questions, and I think there are many flavors to it. I don't think it would be a stretch by any imagination that during the COVID-19 pandemic, early days, a lot of research was done in the MIDD areas. In fact, there have been several publications from the agency side utilizing PBPK, trying to understand whether for some of the molecules that were being considered as potential, to see if they would even have the viability of having the kind of exposures in target organs, which would invoke your typical PBPK kind of approaches, where there are no ways of sampling in organs such as lungs and things of that nature. So there have been lots of applications. In fact, in my presentation, almost all the regulatory review impacts that I talked about, they had significant impact as it pertains to an pandemic kind of situation. Either they decreased the stay in the hospital purely based on using MIDD approaches to support those kinds of dosing regimens, or support longer interdosing intervals so that you decrease the exposure in a clinic kind of settings to go and get the infusions for the patients, or even shorter infusion durations such that the presence at these settings where there could be an interface with other patients, or those kinds of opportunities to potentially get infected are decreased. So there have been lots of applications. So I wouldn't just limit to one or two kind of applications. Obviously, it impacted trials in significant ways, and people have used various MIDD approaches to be able to fill the gaps or interpolate between the pieces of information to be able to ascertain that we did not significantly have a loss of information or end up with uninterpretable information. Now, these depend on a case-by-case basis. Some losses of information can be bridged, others cannot be. So it's not a universal kind of approaches, but most of the MIDD approaches have potential in these kinds of settings, even including trying to figure out what might be the anticipated exposure changes in certain specific population subsets and situations of that nature.
Great, thank you, Raj. There's another follow-up question for Viera. How could our collaborator get access to the DDI standard compounds, and what's your thoughts and input on that?
So they are still general way where the users and collaborators could get access and automatically download any of the models that we have available. Until we finalize that approach and that process, the easiest way is reach out to anybody here at Simulations Plus, whether you do it through our website, through the info page, or reach out to anybody that you know, and we'll discuss whether the model that you need is available, how long it might take for us to build it if we don't have it yet, or if it's available, we'll provide it. So right now it is just reach out to us, and we'll do what we can to get the desired models to as quickly as we can.
Great, thank you, Viera. The next question will go to Beth. So I think you very quickly mentioned that the examples you showed that was following so-called an RTOR pilot program, which is real-time oncology review. And so basically the experience for this project and maybe other opportunities working with the different sponsors, what is observation of the impact of pharmacometrics analysis in terms of timing of the submission in the more rolling fashion? And if you could talk about that, that would be great. Thank you.
So over the past few years, pharma sponsors have approached us, engaged us in helping.
Beth, could you get closer to your microphone? Okay. Thank you.
So over the past few years, as pharma sponsors have engaged us to provide pharmacometric analysis support for their program that's nearing submission, they've alerted us to the fact that the program was on the RTOR path. And so having seen this a few times, I think we're starting to see a pattern. First off, usually we would meet then with the pharma company and very carefully and collaboratively think about the timeline, because generally it's compressed even further than what we normally expect. And so from our perspective, we want to make sure we understand, what's needed, what's the timing of it, and make sure we have appropriate resources and flexibility. So the long and the short of it is usually what's involved is there's kind of a short list actually of what's going to be needed for submission early. And we can use the population PK analysis as an example. And generally, as soon as the model would be developed, for example, you need to create a selected list of summary tables and graphical displays that represent the model or represent, for example, results of simulations that may have been performed. Then certainly also the analysis data set needs to be ready. And so really, at the end of the day, what ends up being different is it's not so much that any of this content you're creating is different than what you were going to do anyway. It's that you need to do it so quickly, as soon as you can, once you have that model run. And so actually, it ends up that you get that work done earlier, and, there may be additional questions and, review feedback or things like that, but it feels like there is a certain pattern to it, and once you've been involved in it, I think it's kind of straightforward.
Great, thank you. And Raj, you're currently with the agency, and Sandra, you've been in the FDA in the past, and any additional thoughts and comments regarding this kind of new initiatives, and what is observation and advice to our friends in the sponsor side, and anything you could add?
Rather than adding, I like to ask if Raj,
Okay.
may I? So
Sure.
Raj, in your presentation, you mentioned that the agency is making a lot of efforts into gathering more resources so that the number of the MIDD submissions can increase, right? And you also mentioned, Raj, that the submissions that are selected are based on the highest impact that can be provided. So I don't know if you have a statistics, Raj, but when it comes to all these submissions, what are the ones that are selected the most when it comes to provide that input? And could you elaborate on that? And also, because I'm close to my heart is PVVM. Have you ever received a submission MIDD that has to do with the application of PVPK, PVVM in support of drug product quality? Thank you so much, Raj.
Yeah, I think you had try to break them down, probably the ones which is most easiest for me to respond to. One of the questions was, how do we go about selecting which ones to go forward, right? The selection is based on several aspects, create a rubric and present it to the cross-center selection committee, which I mentioned, which actually reviews all the proposal and takes in various salient points for each of these meeting requests. And these people, they see various submissions and activities in different therapeutic areas and the needs and things like that. And collectively, the committee together puts in what is the prioritization. So what are the elements that they generally look upon? I think some of the things that they look upon are things like acceptability of an MIDD approach. Do we have past experience, not just in MIDD program, but previously in any other settings, that there is precedence such that we can make sure that we can consistently apply such an approach. Expertise and familiarity. Do we have people who can actually perform? So, for example, artificial intelligence, machine learning are the new things that are up there, and there might be a very complicated artificial learning kind of an approach that someone might bring to the table. But at the same time, we should have the ability also with expertise to be able to understand what is being done, how to go about it, and things of that nature. So we also want to make sure that we don't get into spaces where we don't have an ability to actually evaluate the kinds of complex quantitative approaches that are being proposed, and are they within the realms of what we have seen, for example. So the second is expertise and familiarity. The third aspect that we see is novelty. What are the novel features here? And novelty can come in multiple different ways. It's a unique application to begin with, and does it on its face value represent some meaningful advancement for the drug development program? Novelty can also come in, this approach is being typically used, let's say, in one therapeutic area, but has never been applied in another therapeutic area, for example. And they might not be familiar, the key stakeholders and decision makers, about that. Their historical practice might be that you do these kinds of studies. Maybe there is a way to overcome that or supplement that with some MIDD kind of an approach. So we also see whether we can actually make advances in other areas where there is not uptake of some of these approaches. And lastly, in general, we think about the potential impact. Are we talking about a very big impact to an upcoming phase 3 study, or a critical impact to instructions of use, or with long-term conduct of studies? Now, these become subjective, and that's where we get insights from the senior leadership who sits at the table based on their experiences, and use the collection of these to be able to make prioritization for any given quarter at hand. So that's in a nutshell on how we go about. It's just not limited to these. These are at a high level readily that I can think of. I'm pretty sure depending upon individual cases, we have other considerations also. So that's how we go about doing this. Now, as I gave the answer, I forgot the two questions you asked the other two. Can you remind me?
It had to do with the applications related to to the use of PBPK in support of drug product quality decision making?
Yeah, I do not recollect off the top of my head that we have had specific applications on the product quality aspects that have come into the MIDD program. These have predominantly been with respect to the conventional regulatory decisions, whether we can select this dose or we can select this study in this population, or is the duration sufficient, or can we actually use modeling to predict what might happen in these kinds of instances, let's say for drug interaction situations, things of that nature. But we haven't seen that come through the program. Now, to be fair, under PDUFA 6, there were 3 specific areas, and predominantly, as expected, people came there. Now that we are opening up, maybe we might see it, but so far we don't have experience, at least through the program.
Thank you so much, Raj.
Thank you very much.